There are several excellent articles and videos on CRISPR, but none that make the topic easy to understand for pre-college students with high school level biology knowledge. I poured through many articles and watched several videos to gain a better understanding of the science behind CRISPR. This video is my effort to present CRISPR in a simple way so that high schoolers are introduced to this amazing technology. Some amongst us high schoolers who were blown away by this nifty tool will obviously dig deeper into this technology and may even pursue a career with CRISPR-related technologies. That, in itself, would be a reward for me, knowing that this video may have led them down the path. CRISPR is like scissors to cut something, a pair of molecular scissors. The something that CRISPR cuts is the DNA. You cannot cut the DNA by looking through a microscope, so how is it done? My motivation for preparing this video are summarized in this slide. There is no human inventor of CRISPR. That credit should go to bacteria. As we will see shortly, CRISPR is part of the bacteria's immune system. CRISPR has its origins in bacteria DNA. This is the bacteria's DNA. Prokaryotes have circular DNA. By opening up the DNA, we can see the details. There are nucleotides that make up the DNA. The sequence consists of A, T, C, and G. We notice that there are short repeating sequences spaced apart at regular intervals by the spacer sequence. Note that each spacer sequence is unique. Scientists initially thought that the repeats are significant. Turns out that the spacers are the ones that are more meaningful. The repeat sequence is illustrated at the top. The DNA is double-stranded. Since one strand is 5' to 3' and another is reversed, the nucleotide sequence reads the same from front to back. Just like the word madame, which reads the same from left to right and right to left. This is called a palindrome. So, these are short palindromic repeats, SPR, the last three letters of CRISPR. The spacers are unique, regularly interspaced. The whole thing is a cluster, so clustered, regularly interspaced, CRI. Combining, we get CRISPR. One scientist noticed that the spacer sequence is the same as bacteriophage, virus DNA. Can see here by comparing the nucleotide sequences on top with the one on the bottom. In the bacteria DNA, there are also Cas molecules next to the CRISPR array. Cas is an enzyme. It is a nuclease that cuts DNA. When a bacteriophage infects bacteria, it injects a viral DNA into the bacteria. The bacterial immune system fights the virus by recruiting a combined protein molecule, Cas1 and Cas2. These are a nuclease enzyme, which cleaves or cuts the viral DNA. The bacteria then takes a section of the cleaved DNA and incorporates it into its own DNA. This is the spacer. The different colored spacers shown represent a history of viral infection. Each time the bacteria keeps a small part of the viral DNA as trophy for adaptive immune memory. When the viral DNA is chopped, it gets destroyed. The bacteria lives. Next, the bacteria transcribes its spacer DNA into RNA, or called CRISPR RNA. The nucleotide sequence of the repeats folds upon itself and forms a tracer RNA and attaches to the CRISPR RNA. Tracer RNA provides support to CRISPR RNA. Together, they are called guide RNA, gRNA. The guide RNA is encased in another nuclease enzyme called Cas9. This complex is called CRISPR-Cas9 complex. Next time, when the same virus infects the bacteria, it sends the CRISPR-Cas9 complex for that virus to meet the viral DNA. The CRISPR-Cas9 complex scans the invading viral DNA by opening up the DNA strands. When it finds complementary nucleotides, it attaches to one DNA strand. Only then, the Cas9 protein will cut both DNA strands. The viral DNA is then inactivated and destroyed. This shows all the steps described individually in one slide. First, cuts viral DNA by using Cas1-Cas2 enzyme and incorporates a section into bacteria DNA. Second, transcription into RNA, called pre-cRNA. Third, cRNA plus tracer RNA plus Cas9 forms CRISPR-Cas9 complex. Fourth, if virus reinfects, the right CRISPR-Cas9 complex meets the virus, scans for complementary base pair, locks, and Cas9 cut both DNA strands. The viral DNA is destroyed. CRISPR is very precise. It will cut the DNA only when there is a match, or called complementary base pair. This is important. If CRISPR cuts the DNA willy-nilly, it will cause mutations in the DNA, which can be harmful. Two scientists understood the importance of this molecular scissors that can cut the DNA strands. They eventually received the Nobel Prize in Chemistry for this discovery in 2020. Why is it such an important tool? The entire human genome has been mapped and well understood. So are there many mutations whose nucleotide sequences are well known. There are many genetic diseases which are caused by mutation of DNA, like sickle cell anemia, muscular dystrophy, Huntington's, etc. One can design the guide RNA, gRNA, to match nucleotide sequence next to the mutation. This gRNA can be synthesized in a laboratory with 20 bases long and introduced into a patient. When the designed gRNA finds the complementary base pair in the patient's DNA, it will attach to one DNA strand, like UGA, which is an RNA, with ACT, which is a DNA. Cas9 enzyme will then cut the DNA ahead of this matching base pair, which will happen to be at the mutation region. With mutation destroyed, the disease caused by the mutation will also be removed. The first patient to be cured of sickle cell anemia using CRISPR-Cas9 is Victoria Gray in the USA. Mutation for sickle cell occurs in HBB gene in chromosome 11. GAG mutates to GTG. GAG codes for glutamic acid. GTG codes for valine. So, mutation results in the amino acid valine being produced instead of glutamic acid. This is the nucleotide sequence of HBB gene in chromosome 11. The start codon is ATG. The mutation happens in the sixth codon after the start codon, which is GAG. GAG mutates to GTG, which causes sickle cell. There are two types of hemoglobin, fetal and adult. Only adult hemoglobin can have sickle cell gene. Up to 4 months after birth, our hemoglobin is fetal type. Then, a gene switch in chromosome 2 called BCL11A switches on, and fetal hemoglobin production decreases while adult hemoglobin production increases. As adults, we have adult hemoglobin, which can carry the sickle cell gene. When the gene switch, HBB in chromosome 2 was cut by CRISPR, the off switch became inactive. Fetal hemoglobin production restarted while adult hemoglobin decreased. Cas-GAY gene therapy treatment approved by US FDA in December 2023 for sickle cell and in January 2024 for beta thalassemia. The first patient was Victoria Gray. The patient was completely cured of sickle cell anemia. Alpha globin is common in both fetal and adult hemoglobin. Gamma globin is additional in fetal hemoglobin and beta in adult. 4 months after birth, a switch flips on, which starts to produce beta globin and reduces gamma globin. Beta globin carries the sickle cell gene, but gamma does not. If the switch is removed, gamma globin starts to form and beta globin reduces. This cures sickle cell. Other uses of CRISPR. When DNA is cut, the cell tries to repair, but in the process, there can be mutations. We can deliberately introduce a mutation at the gene whose expression we want to study by cutting DNA. Due to the mutation, the phenotype will be altered. So, researcher will know which gene is responsible for which phenotype. This is gene mapping study. Scientists can introduce an engineered gene in the patient to correct the mutated ones. After CRISPR-Cas9 cuts the DNA, the engineered gene gets inserted in the cut region. Such therapy can cure MD and Huntington diseases. Some companies have lab kits and learning resources for high school students to experience gene editing. Some of the company names are listed. Good way to get practical experience and exposure to lab techniques. Can be expensive. Some regions have limited shelf life and must be transported under cold storage. I hope that you are also impressed with this amazing tool and are encouraged to take up further studies to learn more. Thank you.